Hello, and welcome to the next little super mini lesson of the cell functions video. This one's going to be covering protein synthesis. So before we get into the two processes, which are called transcription and translation, I just wanted to lay some groundwork because some of you may not be comfortable with this concept of what we're about to get into. So we just got done taking a look at mitosis, which is the division of the cell. We saw a little bit about the structure of DNA. And so we've talked about DNA being the blueprint of the cell, carries all the instructions for the cell, but what does that actually mean? And so this process of protein synthesis is going to help explain the role of DNA and actually what it does. In that, it carries the instructions to make RNA, which then carry the instructions to make proteins. So this is what we call the flow of genetic information. So DNA is the storage of the information. Okay, so it's going to store the information. The RNA is a temporary copy of that information, and the protein is the product. This is the genes expressed. So let's take an example. Let me use hemoglobin, right? So we should be comfortable or may have heard that hemoglobin is the protein that's inside of our red blood cells that carries our oxygen. So hemoglobin is a protein. It's a product. Well, how do we get there? So just very schematically, before we jump into the details, let's say we have a piece of DNA. Okay, and on this piece of DNA, we have all of our little bases, A's, T's, C's, and G's, in a very particular sequence. And there is a specific part of this DNA that is going to be the hemoglobin gene, right? So it's going to hold all of the information to make the hemoglobin protein, which is our product. So what happens if we need hemoglobin? So let's say we just gave blood and our body needs to replace those blood cells, and we go to the blood donation place, we give our blood, and your body's like, hey, we just lost some blood. We need more red blood cells and we need more hemoglobin. So a signal will go into our bone marrow, which is inside of our bones, our red bone marrow, and it will tell these cells, these bone marrow cells, to say, hey, you need to make more hemoglobin. So a signal will come in, and we will make a copy of just the part of the DNA that has the instructions for hemoglobin. Oops, RNA is single-stranded. So now we have the instructions just for hemoglobin, not everything, because each cell has the instructions for every single protein your entire body needs. In this particular instance, it just needs the copy for hemoglobin. And so then what is happening is that little piece of RNA, it's called mRNA, leaves the nucleus because it's made a copy in the nucleus. This temporary copy goes and lands on a ribosome. Remember, ribosomes were sites of protein synthesis. And in that process of what's called translation, we get a whole bunch of amino acids, which is what proteins are made up of, right? So the sequence of bases coded in the DNA that was copied in the RNA gets translated in this very specific sequence of amino acids, which will eventually fold around and give us the protein of hemoglobin. Okay, so that's the big picture, that's the big idea. So now we're going to go into these processes, transcription first, which is making a copy of, taking the DNA and making this temporary copy, and then translation takes that temporary RNA copy and translates that into the protein. So DNA to RNA to protein. All right, so let's take a look. So that first process in protein synthesis is transcription. So I just have three separate pictures showing you the same thing. So whichever one kind of fits with your brain a little bit better. So this first one here, what I have is the purple double helix kind of is being pulled open right at that very specific region of the gene. So we can call that our hemoglobin gene. And if you'll notice, we have a light purple, kind of a pink copy coming off of it. And this is what we call mRNA, where the M stands for messenger. Is the same thing as mRNA, okay? because it's carrying the message from the nucleus out to the ribosome. But if you take a look at that mRNA strand, you might notice something a little bit different. So remember our 4 bases were A's, T's, C's, and G's? Well, if we take a look at our RNA piece that's coming off, we have A's, C's, G's, but we have U's instead of T's. So it's just a little bit different. RNA is a little bit different than DNA in that instead of having thymines, it has what's called a uracil instead of thymine. But it partners with the A just fine. So wherever there is an A in your DNA, the RNA would put in a uracil. But it's going to function exactly the same. The ribosomes are going to know exactly what to do with it. So if we look at the next picture, kind of down here, we see the same thing where the DNA is kind of the orange backbone. And here's its bases, the A, T, G, C, T, A, U, G, G, C. And then an enzyme is going to come along and make the mRNA with those complementary bases. And then lastly, we have the same kind of picture over here where we have our DNA double helix being pulled apart and our complementary mRNA being built. Okay, so this is the process of transcription, takes place in the nucleus. We are making a temporary copy of just the one gene of interest that we are wanting to make the product for, and that is our mRNA, and it eventually will leave the nucleus and go land on a ribosome, and that's what translation is. So in this particular diagram, this came from another resource, so I don't know if you have this in your textbook, but picture one, or number 1 here showing transcription. So you can kind of see that blue temporary copy, that's the mRNA coming out from the DNA, leaving one of those nuclear pores, coming around and landing on a ribosome, the orange kind of sandwichy thing. So what we're seeing happen in translation is each one of these little key looking structures, this guy right here, this is called tRNA, which stands for transfer RNA. And what tRNA does, its job is to transfer the correct amino acid from the cytoplasm to the ribosome so it goes into that very perfect order to make your protein product. And how does it know which one to grab? Well, we have these 3 bases, right? So it's kind of highlighted 3 here, and it's going to have the next 3 here. Every 3 bases codes for 1 amino acid. Every 3 bases in the mRNA. So let's pick methionine just because I know that one. So there are 20 amino acids that are all of the ingredients to make all of the proteins we need in our body. And if we have our amino acid methionine, if we need that one, it's not letting me draw an E. Let me try a little bit bigger. M-E-T, there we go. So if our amino acid is methionine, and that's what we need to bring into the ribosome, our mRNA sequence should be A, U, G. Like, Corey, how the heck did you get that letter sequence? Because if we look at this code here, the first base in the codon is A. And if the second base in the codon is U, right, that's going to put us in this column. Okay, so we're in this row. And now we're in this column. And G is the third base in the codon, means we have to be on this row, this smaller row, so that puts us at methionine. So if we have an AUG coming in here, these tRNAs know to bring in methionine as the next amino acid in that DNA or in that protein sequence. So let's see if you can figure out the next one here. Oh, I don't remember. I don't know the code numbers on these. So let's just do, I'll just pick one. What if the codon was UGG? So we have UGG right there. So the very next tRNA has to recognize UGG. What would be this amino acid on that tRNA that's recognizing UGG? Well, let's use the genetic code and figure it out. So if the first base is a U, it's got to be somewhere in this row. If the second base is a G, it's got to be somewhere in this column. And where those two intersect, we have cysteine, cysteine, stop, or tryptophan. So then our final base is G, so then it has to be in this row here. So the answer is tryptophan, TRP. So this very next amino acid coming into our chain for hemoglobin is going to be tryptophan. And it goes through that cycle the entire length of the mRNA until your protein is completely done. And then it pops off the ribosome breaks apart, the mRNA breaks apart, it goes and gets recycled. It does not stick around because it's temporary, right? And so that is how we get our protein product. So from DNA to RNA to protein. This is what we call protein synthesis. All right, so that wraps up our cell function lesson. The next video is going to kind of be our transition into tissues, where we're going to take a look at a whole bunch of different types of cell diversity. And then the last part will be covering all of our histology. See you next time.